Hi, my name is Tanya, and this is the presentation of my poster on biosynthetic gene clusters and protein language models. Many organisms, such as bacteria, fungi, and plants, produce intricate chemicals that are not needed for the growth and reproduction, and thus are called secondary metabolites or natural products, or NPs. NPs are a rich source of drugs, with most antibiotics being derivatives of NPs. In a producer organism, NPs are synthesized by a set of enzymes encoded by genes that often lie near each other on the chromosome and are called biosynthetic gene clusters. Despite the clinical importance that some NPs have, only a small number of naturally occurring BGCs is explicitly described. In cases like these, the rule-based solutions and probabilistic models, which don't require a lot of data, work best. We don't know anything except those rules. It's hard to generalize, it's hard to predict the new BGCs de novo and characterize their products. The biggest publicly available dataset, which has rich annotations for BGCs and corresponding natural products, is called ECCB. The version we currently use in this work contains information of 2,500 of biosynthetic gene clusters. As you can see at the upset plot, which describes it, some of the BGCs in ECCB are annotated with several different biosynthetic classes. We exclude them from the further analysis, thus having 2,000 of BGCs left for experiments. The total number of samples we have is quite small, but we can still use deep learning there. And the protein language models might help us here. Typically, these models are trained on general tasks like mass language modeling task, where the model is trained on to predict a missing amino acids in a protein sequence. And typically, the dataset for the training is huge. These models produce vector representations, so-called embeddings, on amino acid level or on a whole sequence level by averaging the amino acid embeddings. The table illustrates some examples of state-of-the-art models and with their performance after fine-tuning for several downstream tasks. Looks nice, hey? For our dataset of 2000 BGCs, we'll use protein language model as a backbone of a CMS network. The scheme of the typical CMS network is shown on the slide. Our input is a pair of protein sequences corresponding to a pair of BGCs. The network computes a pair of embeddings for these two sequences and computes cosine similarity between them. We can fine-tune the network to morph the embedding space to make the cosine similarity between embeddings close to some user-defined metric. One of the cool features of CMS network is that they are capable to provide nice results even in cases where the training data is very limited. That's exactly our case. In this work, we'll try to solve two different tasks with CMS networks. Let's take a closer look. In the first task, we try to predict if the two BGCs belong to the same biosynthetic class or not. We try to solve this task with two different dataset split schemes. The one simply tries to keep the percentage of samples on each class similar in all splits. Another simply considers one of the 7 biosynthetic clusters to be in our test dataset and uses for training validation everything else. First, we wanted to make sure that our model actually learns something. The first PCA plot shows the embedding for BGCs, which were computed by pre-trained model with no fine-tuning. After that, we trained the model in two different settings. Here you can see how our latent space have changed. Note that both pictures looked similar, despite the fact that the model which produces the embeddings in the further picture below didn't know anything about NRP class. We also tried to look at the cosine similarity distribution. The model which we used to compute similarities for the left plot was trained with no information about the RIP, yet it can separate them from the other classes. Embeddings on the right plot were computed by the model which had no information about NRP class. It doesn't separate NRP from perlitide, which is okay because these two classes are similar in terms of domain organization. To evaluate the results of our experiment, we computed the silhouette scores of our models. As a baseline to compare, we took the big escape distance matrix. We filtered the BGCs to be present both in the escape results run and in our model's test datasets. What can we say about this experiment? First, the fine-tuning worked and improved by synthetic class separation. The fine-tuned model performed well for the classes they haven't seen and might be used for de novo prediction. As a preliminary work, we tried to compute animated similarities between all pairs of fingerprints belonging to a pair of natural products produced by BGCs within the same biosynthetic class. In the plot, you can see that the Nimote similarity between NPs differs within each class. This means that the dataset in terms of NPs is quite diverse. In addition, the biosynthetic classes themselves have the similar dominant organization. We decided to simplify the task by focusing BGCs belonging to the only one class, NRP. As a result, we are dealing with an extremely low resource dataset here, 228 samples in total. Despite that, after fine-tuning, our model learned something that could predict an Nimote similarity between the natural products given the sequences of the corresponding BGCs. In this task, we also use the same big-scale distance matrix as the baseline and compute the correlation coefficient between the metric predicted by our model and the corresponding distance. The negative signs are fine, since we compute the similarity with the metric here. When the similarity is high, the distance is low, and vice versa. In this experiment, the model was capable to predict chemical similarity within one NRP class. We plan to try to use other multimodal data as a target metric in the future. Thank you for your attention. Future project updates will appear at our GitHub repo.